Good morning, good afternoon, and good evening, and welcome to our webinar, Reduce Errors and Increase Confidence in Testing with Quality Controls. My name's Joe, I'm your host, and I want to thank you all for taking time out for what will be another very informative presentation from the experts here at Antilia Scientific. Now, before I introduce you to the wonderful Jessica Van Allen, I'm going to run you through a few key items just to get you settled in today, okay? So, first of all, you're all viewing this, and you should see lots of floating boxes. All those boxes can be moved around to suit your own screen resolutions. So we fully appreciate that not everybody has the same size monitors. You might be watching this on an iPhone or another smartphone. So you can rearrange your viewing experience. And we urge you to make sure you get settled in and nice and comfortable so you can see the slides. Hopefully you can see me. But more importantly, there's a Q&A box. So hopefully you can see that too. Now, obviously we want you to ask questions. All the questions are anonymous. So as you ask them, no one else can see what you're asking. And equally, sometimes we do get a chance to answer those questions, but if we do answer them, we'll type the answers out. But if you don't hear from us, what we do is we collate the questions at the end of the presentation. We make a small amount of time to do a Q&A for you. So we'll read out the questions that we've received. Now, please don't worry. If your question is not read out, what we do is we collect them all into a Word document, and a few days later, we'll send that Word document out to everyone here today with all the answers to all the questions that are asked. Okay, so please do ask questions. We really like it when people ask questions. And also, this video is being recorded. This webinar is being recorded for video. So an on-demand link will be sent out to everyone here today, and to all our colleagues who, if any of our colleagues couldn't make it today, a link will be sent out just over an hour after this webinar ends. So again, you'll get a chance to rewatch this webinar and also share it with colleagues as well. And we really do urge that if you find something interesting that you do, please share it with people that might find it interesting too. And there's just one more thing. There's a very short survey, and it's available to do it now, but I appreciate you're all watching me at the moment. So at the end of the webinar, the survey will pop up. So please do just take 30 seconds just to help us help you. Now it's time to introduce Jessica. So Jessica has a PhD in biochemistry. She's been with Zepto for 9 years now, which is a good deal of time. She started as a senior molecular biologist and worked her way through the QC department. And she's very focused on this natural molecular quality control development product. And I'm sure I probably pronounced that completely wrong, but natural, I think is how you pronounce it. She's now a lead technical support specialist, and she's serving customers all over the globe, and we get all sorts of inquiries, and she does an amazing job here. I'm sure Jessica won't mind me saying that. I'm trying not to embarrass you too much, but on that note, I'm now going to hand you over to Jessica, who's going to talk you through the learning outcomes of today's webinar. Thank you.
All right, thanks so much, Joe. So as you saw from the title slide, today we're going to be talking about how we can reduce errors and increase confidence in the testing that you're doing in your laboratories with quality controls. So I'm going to start by doing a brief overview of molecular diagnostics and kind of the field at large and how we got to where we are now. And then we're really going to dive into quality controls, why quality controls are important, the roles they play in our laboratories, and namely what makes a good quality control. And then lastly, I'll spend a couple slides talking about how I believe Zeptometrics offers a total solution for quality control materials in your labs. So as you can see, this is a pretty, truncated version of the evolution of molecular diagnostics, but I think it's helpful to kind of put us into context today to see where we came from and where we're going, and the evolution of these processes. So if you've opened a molecular biology textbook anytime in the last 10 to 15 years, 20 years even, you probably know all this, but prior to the PCR, the diagnosis of infectious diseases was really laborious. It was expensive, and it wasn't especially accurate. In the late 1980s, with the establishment of PCR and the start of things like the Human Genome Project, these advancements came really quickly with this wealth of information that was being generated. And by the late 1990s, RNA amplification methods were being used specifically to diagnose HIV, and tests for things like HCV and CMV and EBV followed very shortly after. And around the same time, DNA amplification methods were being honed. And HSV, I think, in cerebral spinal fluid was one of the first. And around this time, also, the first FDA-approved molecular diagnostic testing kit was released for the detection of chlamydia trachomatis. Of course, along with these, immunoassays were also being developed in tandem. And though multiplex PCR had been around for several decades prior to this, it really didn't rise to prominence until the early 2000s. Obviously, using multiplex PCR panels help meet laboratories' needs for high sensitivity and specificity and quicker turnaround times. But these methods were also typically beneficial in terms of cost for training of laboratory staff and to really implement uniform workflows in the laboratories. Importantly, multi-pathogen panels allowed the co-detection of species that had previously been incompatible to grow in the same culture systems. So you can see how this kind of testing was really a game changer in the field. So with the completion of the Human Genome Project in 2003 and the technologies like next-gen sequencing, high-throughput sequencing, there was really an incredible amount of data and information being generated and gathered at this point in time. So this massive influx of information and the need for labs to quickly and accurately diagnose infectious diseases really spurred on the development of these point of care systems that we're familiar with today. And obviously that area continues to push forward with brand new innovations. Syndromic panels, those that look at bloodborne pathogens, GI, respiratory, tuberculosis, just to name a few, not only can we now test for these diseases themselves, but we can now directly identify important antibiotic resistance markers. The impact of this, you could go on and on talking about. So obviously this is not an incredibly in-depth look into the field at large, but I think the point I want to impress is that the advancement of molecular diagnostics has been relatively fast, scientifically speaking. And I think all of that leads us to today, where we're at the point that it's possible to characterize a new virus, in the case of SARS-CoV-2, sequence it, and develop point-of-care tests in a matter of months, which is kind of incredible. Obviously, brand new discoveries are being made more and more often, and what we're seeing now is that the time is shrinking in between each new innovation. So really now the race is to further increase sensitivity and specificity always, to cut costs, make it more affordable, to make these point-of-care instruments especially smaller, decrease their footprints, make them easier to use, and the top priority being to decrease the time it takes to get a result, to go from a patient sample to a diagnosis. So I really like this quote. It caught my eye when I was putting together this presentation. If you'll allow me to wax poetic a bit about molecular diagnostics, it says, "Medicine is science, experience, and art. Obviously, medical and clinical laboratories provide a major source of information about the patient to the practitioners. So therefore, as we know, the accuracy of the data and their interpretation is critical." Clearly, molecular diagnostics have come a long way over the last few decades, and especially I think in light of the pandemic, they've become a gold standard in clinical laboratory medicine really around the world, especially in the case of infectious disease diagnostics. These molecular tests have to constantly adapt and evolve to meet the present needs. So despite these dramatic advances in laboratory technology, many patients still continue to receive inaccurate, incomplete, or delayed diagnoses that can result in suboptimal treatments and outcomes. An analysis that was performed by the Infectious Disease Society of America found that the direct costs for care of patients suffering from infection were in excess of $50 billion annually. And I think that it's possible with more rapid or more accurate diagnostic tests that more lives could be saved and these healthcare costs could be drastically decreased. I believe that no situation has really put this into a bigger spotlight than the current SARS-CoV-2 pandemic. And not only do we need accurate, quick, reliable diagnostic tests, but the labs performing the testing must have a way of monitoring the test performance so that they can catch any errors or any issues with, say, reagents or test operators and various processes that are going on in the labs. I think whether you're a molecular lab who's trying to develop or are currently implementing a laboratory developed test, or your clinical laboratory who's running patient samples, I think the quality of your procedures and of the instrumentation you're using is really of the utmost importance. So like we talked about, molecular diagnostics are an integral tool in catching these infectious diseases. And infectious diseases are still a major cause of death around the world. Lower respiratory infections were the world's most deadly communicable diseases. They were the fourth leading cause of death, according to the WHO, from their 2020 report. And clearly, right now, over 5 million people and counting have died from COVID worldwide. Now, it's not all doom and gloom. Obviously, we made a lot of advancements in this field. Specifically, if we look at the incidence of HIV, tuberculosis, and malaria, those have declined, with death rates attributable to them decreasing annually by roughly 3%. So that's great. And I think we see that because we're getting a lot better about diagnosing, treating, and preventing these infectious diseases. So, but those, along with other tropical diseases and other GI diseases, still remain major killers of those in middle to low income populations. I think while it's easy to think in terms of infectious diseases or causes of death in the context of our own communities, global health is and really should be a major focus. Just because an infectious disease isn't currently prevalent where you live doesn't mean it can't be. I think that's kind of highlighted right now. Good global health at the end of the day is good for everyone. Really, that could and should be a separate presentation in and of itself. I think the pandemic brought forth this inequality in healthcare and treatment availability as a result of the lack of resources in those low and middle income communities. So I think the moral of the story really remains the same here, that early and accurate detection with infectious diseases saves lives. So we're going to look here at a little bit of data that was generated throughout the pandemic and talk about molecular diagnostics as it relates to SARS-CoV-2 specifically. So this graph shows US FDA emergency use authorizations, or EUAs, that were given out throughout the pandemic. And this is data through September 30th, the end of September 2021. So as you can see, the blue box on the graph, number of tests is on the y-axis, and the blue box is molecular tests. Over 200 molecular tests received EUA from the FDA during the pandemic, by far leading the pack in the United States. You can see antigen and antibody tests also bringing up the rear there. But not only in the US, but also worldwide, we can look at the number of tests that are in development worldwide. And again, molecular leads the pack with about almost half of the test being qPCR based. Again, using those in conjunction with the antigen and antibody tests to really accurately diagnose SARS-CoV-2. So like we just talked about, molecular tests really isn't slowing down. In fact, there was a recent study from the Journal of the American Medical Association, or JAMA, in K through 12 public school systems, and it suggested that screening and environmental monitoring caught far more cases of COVID-19 than the conventional reporting. And conventional reporting in this case, they're referring to either getting tested when you had been told you came into contact with someone who had COVID, or getting tested because you had symptoms. They're more passive methods. And they found as many as 9 in 10 student COVID cases and 7 in 10 staff cases could be missed by conventional reporting mechanisms. So this really highlights the need for a more robust and active rather than a passive testing plan. And I think when you think about this, we have to take on a lab by lab basis the approach of, okay, what is our end goal here? And I think that answer to that question, it always circles back to positive outcomes, be it development of a new assay to test for an infectious disease, or if you're a clinical lab providing patient results. Really unequivocally, the goal is to produce the highest quality result as possible. Yes, specifically in the case right now of SARS-CoV-2, but really for all testing moving forward, that should be the goal. I think with this pandemic, we might have been caught a little bit on our heels in this instance, but we're lucky enough to have the advancements made in this field up to this point that we were really able to adapt incredibly quickly using the existing technologies to develop the tests that we needed. But what I think is a major takeaway here is that more testing equals a greater need for good quality control systems and a need for the availability of reliable quality controls to be used across all test systems and platforms. Of course, different kinds of tests have different kinds of controls. So the purposes of today, we'll be focusing on specifically PCR-based molecular diagnostic tests. So before I jump into the differences of controls, I kind of want to talk about quality management systems a little bit. So your lab's quality management system really provides a framework for you to manage and monitor the activities going on in your lab to address both quality standards and the goals of your organization. I think there are countless considerations that go into these quality management systems that we really won't touch on today, things like purchasing and documentation, personnel. You can go on and on about that. But one chief part of a good quality management system includes process control. And this refers to the control of the activities that are employed in the handling of samples and the examination processes in order to ensure accurate and reliable testing. All quality control processes fall under this process control term. And really the goal of quality control and in using quality controls is to detect, evaluate, and to correct errors due to things like test system failures, environmental conditions, or operator performance before these patient results are reported. And I think if we're talking in terms of quality control, I think sometimes internal quality controls and quality controls, those terms are used interchangeably depending on the situation. I know the WHO tends to use internal quality controls or IQC a little bit more broadly in their definition. But for the purposes of this presentation, when we talk about internal controls, we're speaking about built-in controls or controls that are built into the system. So really, quality controls can be broken down into kind of two families, built-in or internal controls, like I just talked about. And those are integrated into the design of a test system and are performed automatically with each test. Typically, they only monitor a small portion of the test, and they do not monitor for all the conditions that could affect your test results. On the other side, we have external controls. As the name implies, they need to be purchased or made separately, and really their goal is to evaluate the testing process a bit more broadly than built-in controls can. You want them to assess the integrity of your entire test system. Ideally, you want to have both positive and negative controls on board, and you can make them in your lab from stock cultures that you've acquired, or you can buy them from quality control manufacturers. If you're using pooled patient samples, it's important to consider if that's a right fit for your lab because they likely do not meet ISO standards. It's just something to think about. And really, controls are they're substances that contain an established amount of the analyte that's being tested. And really, you want to use external controls to test the same time and in the same way as your patient samples are being tested. Really, what the purpose of a quality control is, an external quality control specifically, is to validate the reliability of your test system and also to evaluate your operator's performance and the other environmental conditions that could impact your test results. So why quality controls? The CLIA program, I'm sure you're well aware if you're in a clinical laboratory, they regulate laboratories that test human specimens and ensure that they give accurate, reliable, timely patient test results. And labs can obviously obtain CLIA certificates depending on the kind of diagnostic tests they perform. And CLIA states specifically that the laboratory is ultimately responsible for the performance of appropriate quality control procedures, really putting the onus on the laboratory to set up their own quality management systems. And they do go on further to say that for each test system, the laboratory is responsible for having control procedures that monitor the accuracy and precision of the complete analytic process. That again brings us back to external quality controls. And then we can look a little bit further and look at ISO guidances. ISO guidances tend to be for quality in manufacturing and service industries, and they have a little bit of a broader applicability. What they say is that quality control materials should be periodically examined with a frequency that is really based on the stability of the procedure and the risk of harm to the patient if they were to receive an erroneous result. And they specifically recommend that with each new formulation of exam kits or changes in reagents or procedures or a new lot of shipment should be verified for performance before use in examinations using external controls. And I think specifically, ISO also points to independent external controls. So the use of these independent third-party control materials should be considered, and they say either instead of or in addition to any control materials supplied by the reagent or instrument manufacturer. And like we talked about a little bit when we talked about external controls, they should use quality control materials that react to the examining system or the test system in a manner as close as possible to patient samples. A rigorous QC program in your lab not only helps to fulfill these regulatory requirements that are set forth by ISO and CLIA, but also it increases your result confidence, right? So it can also enhance your efficiency in the laboratory, and it saves you money. It minimizes the waste. And by and large, the goal of it is to simplify your laboratory procedures. You want to take the guesswork out of what you're doing. So all of these regulatory bodies, ISO, CLIA, CLSI, CAP, really they're looking to make quality control a standardized procedure. Obviously, the availability of molecular diagnostic tests is booming right now, but considering we didn't even have PCR until the late 1980s, is still quite a young field. And I think it's the hope that independent quality control materials will bring molecular infectious disease testing to the level of standardization and uniformity that's needed in clinical testing. If you're relying on solely in-kit controls, you have to keep in mind that those are really optimized to function with specific assays, specific platforms, and reagents. So obviously you can see that it could introduce a little bit of a bias. So it's critical to use well-designed, independent, and unbiased third-party QC controls in addition to, I know they say in lieu of, but really I think ideally you'd like to use them in addition to in-kit controls, especially full process controls, because unlike with synthetic control that might only contain part of the genome, each of the extraction, amplification, and detection steps is fully controlled in an unbiased and thorough manner. So I think besides the obvious advantages of independent external controls, which are monitoring the precision of the results from your test kits and from non-commercial assays, and preventing errors by constant and consistent monitoring, these are a few other advantages that I thought of. Truly independent external controls, that is those not tied to a specific platform, are really advantageous for efficiency. Efficiency because you only have to buy one control to work across multiple platforms or tests. And they offer, like we mentioned, an unbiased performance check. Independent quality control manufacturers have their own quality management systems in place. So when you buy from them, you're ensured quality in their processes and their products when it comes through your door. It can save time. Obviously, you might think you might be saving money by making these controls yourselves. But really, when you think about all the time that goes into it and the effort and the reagents and supplies, you might be better off just buying from a third-party supplier. And lastly, I think that's a really big advantage, is troubleshooting help. If you order from a QC manufacturer, you have a technical support system to back you up. So you're not alone in trying to troubleshoot or work through any issues in developing your assays in the lab. So when should you use quality controls? And we'll look at this really through the lens of SARS-CoV-2 as an example. And kind of the name of the game here, I think, is mixed messages. So in the specific case of SARS-CoV-2, the WHO recommends that QC testing be performed with each run. And they note that each NAT run should include both external and internal controls. And labs, of course, are encouraged to participate in quality assessment externally as they become available. So that's what the WHO says. If we look at what CAP says, they state that for molecular-based quantitative and qualitative tests, controls are run at least daily or more frequently if specified in manufacturer's instructions. So I think when to use quality controls depends really on how you're using them. You know, quality controls are versatile. You might use a different set of controls, say a quantitative set, when you're establishing a new test in the lab, or you might need something like a verification panel if you're onboarding a new platform in the lab. And then, of course, once you have that platform established and it's passed its verification, you might want to establish a consistent QC regimen with some kind of qualitative pooled control or an external run control to cut down on the number of runs needed. So really, like we talked about a few slides back, it is up to the individual laboratory to generate and to stick to their quality control plan. So your QC plan really should include considerations like manufacturer's instructions of the test that you're running, the quantity of tests being performed in the lab, and how often reagents need to be ordered, for example. That's just a few of the considerations. Like we talked about a little bit earlier, CLIA regulations require a lab to have QC procedures in place to monitor the accuracy and the precision of the complete testing process. And they actually do describe a relatively new option that's available, which provides laboratories the opportunity to tailor what they call an individualized quality control plan, or an IQCP, for their specific testing environment and for their patients. And this option actually offers a lot of flexibility for meeting the regulatory QC requirements that are appropriate for the testing that's being performed. IQCP really should be as restrictive or more restrictive than what the assay manufacturer provides. And you can find a step-by-step guide for developing an IQCP on the CDC website if you don't already have one in place. I think here are some common instances in which you should consider running external quality controls. The idea behind using an independent quality control is that the material is multifaceted, like I just talked about. If you buy from a QC manufacturer, you'll likely able to find a quality control that can meet your needs across many of these testing instances. So performing a verification on a new assay or platform, performing a validation, before and after instrument maintenance is a really good time to do this. Obviously, when you're changing suppliers or methods for reagents or supplies for an existing assay you already have in place. When you receive a new reagent lot in, you want to be able to assess the variation between different lots of reagents, and hopefully you can observe any shifts in data that might be caused by that. And lastly, regular training of technicians. Quality controls are super helpful in that vein. So what's at stake when you aren't using quality controls? I think the answer is, quite frankly, a lot. I think this Benjamin Franklin quote, it really fits here. An ounce of prevention is worth a pound of cure. Preventing adverse events is far preferable to having to implement corrective actions after an issue arises. More and more, people are looking at what the cost of quality is in the lab. And there's cost of good quality, the preventative measures that you take, but there's also the costs of poor quality that people are really starting to look at. That is costs of physical things like reagents and supplies and having to retest over and over. Time, of course, which really equates to money in both molecular and clinical labs. And in the case specifically of clinical labs, misdiagnoses and delay in medical care because of these misdiagnoses or these late diagnoses. And really, your laboratory accreditation could be at stake here if you don't implement a plan that involves a high quality molecular control. You need to be able to show that your lab can continually perform to high standards. So if we take, for example, consider the cost of a poor quality control program in clinical lab. So if you're a lab running an instrument that gives patient results, say, you're short staffed, you're looking to cut costs, you take your QC procedure from daily QC down to weekly QC. So you're performing it once a week. If in between those 7 days, something changed that affected the data being generated by the instrument, it wouldn't be caught until the following week. So if you think about that, that would put all the data that was generated between those 7 days into question. So I think you can see how costs would really quickly start to add up under that circumstance, not just reagents and supplies, but the time that was wasted. And obviously, if tests were being sent out to a patient, you could have a misdiagnosis on your hands. So what should you look for in quality control? This is kind of a laundry list of things that I think are really important to consider. You want to have a full process or whole organism control, like we talked about. This really allows laboratories to validate the performance of the entire scope of the assay that you're using. Especially in a molecular PCR-based assay, you really want to track the extraction, amplification, and the detection. And by using a full process control, you get all three in one. So it's a huge cost savings. And importantly, you do want to try to find a control that's supplied in a matrix that does mimic the clinical specimen that you're using. Safety obviously is important. You don't want to have to use infectious material when you're generating quality controls if you don't have to. Ideally, you want to have inactivated material, and a lot of them on the market now, a lot of the controls are either synthetic, RNA and DNA, or heat inactivated or chemically activated materials that are much safer to use on the bench. Ease of use is also something to consider. You want these controls to be ready to use without much manipulation or thawing. Traceability, if you're looking at a quantitative control. Shelf life, you want something to last, as long as your reagents are lasting, or ideally even longer so you can compare across reagent lots. And like we mentioned, you want them independent. And you want to make sure that these controls are manufactured under good quality systems. So this is just a small snapshot of the handful of test makers out there who have won FDA EUA approval, like we talked about way at the beginning of this talk, for SARS-CoV-2 tests since 2020. And I think with so many choices and each of these options with a different IFU or instructions for use, it can be really difficult to figure out where to start when looking for quality controls. So for the last little bit of this presentation, I'm going to talk to you about how Zeptometrics natural quality controls really, I think, provide a total solution for your laboratory's quality control needs. I think through our unique patented natural portfolio, you'll find that Zeptometrics really checks all the boxes we talked about a few slides back in regards to what makes a good quality control in terms of full process, safety, ease of use, traceability, shelf life, etc. So really from the onset of the pandemic, Zeptometrics was leading the charge in acquiring SARS-CoV-2 to generate controls. As I'm sure you're aware, the virus itself was isolated in January of 2020, and by March, Zeptometrics had created SARS-CoV-2 recombinant constructs, only to be followed very shortly after in May with a whole organism full process SARS-CoV-2 natural external run control. And basically from then on, Zeptometrics has really been working to acquire relevant strains and variants of interest, and we've provided many of these as heat inactivated or UV inactivated culture fluids. Zeptometrics also carries a number of panels and controls with known performance on common point of care tests and IVD tests, and we work with some of these assay developers to add SARS-CoV-2 into these existing controls and panels to help customers. And we've also provided a lot of custom OEM materials throughout this time period. And as recently as October 2021, Zeptometrics released a SARS-CoV-2 multivariant panel. And really, we continue to innovate and add products as needed. So how does Natural meet your quality control needs? So first, if you haven't heard of Natural, the name stands for Nucleic Acid Test Controls. And it's a patented technology that we developed, and it's compatible with all kinds of microorganisms, viruses, bacteria. And what it does is it renders the material non-infectious and refrigerator stable. Obviously, that checks the box here for safety, non-infectious. You have a whole organism present, so you have your full process control, so it's safe and easy to use. It's refrigerator stable, so that really speaks to the ease of use. Like I mentioned, a lot of controls available on the market are frozen. So with that, you have to really be careful of the amount of freeze thaws that you do, so you don't have to worry about the integrity of the material. Or a lot of them are also lyophilized, and obviously lyophilization requires a reconstitution, and that requires a little bit more care and pipetting in order to ensure that the correct concentration of the analyte is being used. So Natural being refrigerator stable, you can pull it out of the fridge and run it as you need. Really easy to use. And not only that, but being refrigerator stable doesn't require dry ice or any special shipping requirements. So you actually do save on costs there. Also, a lot of our quality controls are test and platform agnostic and are also manufactured to ISO 13485 standards. So really the range of Zeptometrics' third-party control solutions cover an extensive range of infectious diseases, anything from transplant-associated diseases, bloodborne, respiratory, GI, STI. We really cover the gamut here. And like I mentioned, many are platform or assay agnostic. meaning that they're not tied to a specific test or assay. We do also offer quantitative materials that, if possible, are correlated to an international standard. And really, these could be ideal for analytical testing for laboratory developed tests. We do also have a number of controls and panels in a ready-to-use format that are platform and assay. They have known performance on different platforms and assays on the market. And really, we offer a huge breadth of line and depth of line in our materials, meaning, we have a lot of viruses and bacteria and different microorganisms on hand that we can readily turn into natural. We are constantly looking for new and emerging disease outbreaks, and we are constantly sourcing multiple strains and variants of viruses and bacteria. And we have the capability to formulate in various matrices or diluents to do custom and OEM work. And for those organisms that you can't easily culture, we do also have the technology to develop recombinant organisms. And really, Natural is geared towards the laboratories that are carrying out molecular or PCR nucleic acid testing. So this slide really here just shows you kind of how Zeptometrics can help you out at every stage of the process in which you would use quality controls in your laboratory. They're multifaceted. They can be used across a wide range of instances. So I think looking at this, you can see that there's a solution available for everyone. This is just a very basic breakdown of some of the ways Zeptometrics controls can fit into your laboratory setting. We talked about some of these times earlier that you might need quality controls. And I think really the takeaway here is that we truly offer something for almost everyone. And I'm hopeful that if you look at this and notice, oh yeah, I'm doing some R&D in my lab, I could really use some quantitative controls or a linearity panel. We really do have a broad organism offering that can help you. And we talked about validation and verification. Like we mentioned, we have a lot of controls and panels that have known performance on platforms. And then that also leads into doing routine QC, where our external run controls, and again, those panels and controls can come into play. So I'm really hopeful that you'll consider Zeptometrics when the time comes for you to implement your laboratory's quality control plan. So really, like we talked about today, molecular diagnostics is a continually evolving and growing field. And I hope you took away that implementing QC materials as a part of your lab's quality management system really is crucial. And that, if at all possible, independent third-party quality controls should be used in conjunction with your internal controls. And lastly, I hope you see that Zeptometrics can offer a really unique and reliable solution for independent quality control in your laboratories. And that's all I've got.
Brilliant. Take a breather, Jessica. You did really well. Thank you so much for that informative presentation. Yeah, so it's Q&A time. So yeah, have a drink, get yourself ready for this. Okay. So remember what I said earlier, if your question is not read out, please don't worry. We will put all the questions together in a Q&A document, and it will be sent out to everyone here today. So Jessica, first question, is a control different from a calibrator?
Sure, yeah. I think sometimes these terms are used interchangeably, but typically, when we talk about a calibrator, you're talking about a solution that has a defined or specific concentration that's used to set or calibrate an instrument or a system before testing is started. And a lot of times those are provided by an assay or instrument manufacturer. And really how that differs is that a control is really meant to validate the reliability of the test system itself. So it's run with patient samples and in the same method as patient samples. And that's more to evaluate outside factors like we talked about, like operator performance or different environmental conditions that could influence results. And really they should mimic patient samples so as to challenge the full process of the assay in question.
Yeah, that's a really good answer. Thank you so much, Jessica. How often should controls be run?
I think like we talked about in the presentation, it's not super clear. And I think that's on purpose. These guiding bodies want you to set your requirements based on what your laboratory means or what your laboratory needs and the testing that you perform. So really, if you should follow your laboratory's IQCP that's established, it permits your lab to customize your QC plan according to the test methods that you are using and the environment and the personnel and the competency of your personnel that you have working in your lab. Um, you always want to look at the reference the manufacturer's instructions to the assay that you're using. They can often give you a little bit of guidance there. But like I mentioned, you can go to the CDC website if you don't have an IQCP plan in place. It's something that's really, really crucial, and every lab, I think, should really work towards.
Perfect. Thank you, Jessica. How do I know which kind of controls I need for my lab? And it goes on to say that is qualitative versus quantitative, or panel versus controls. Yeah, I wonder if you can answer that one.
Yeah, I talked about that a little bit at the end, didn't go into too in depth for it. But the kind of controls you need obviously depend on the tests that you're running in your lab. And if it's a laboratory developed test, if it's an IVD test that's on the market. And really, the people at Zeptometrics, our technical experts, would be happy to work with you if you specifically have questions or you're using a specific test and you're unsure. We're really happy to work with you and answer your questions in determining which products might align best with your laboratory's quality control plan. So please contact us.
Perfect. Yeah, it's a great message there to end with. So, that's another good point. So thank you so much, Jessica. I'm going to end the Q&A there. There were a few more, but we're sort of running low on time now, and I think it's best that we just wrap up this webinar. Jessica, if you have anything specific, she can be contacted directly on her email address, which is on the screen right now. There are a couple of tools around the webinar. If you want to request some more information from the Zepto team, there is a little box there that says request more information. Just click on that request now button and just pop a note over, and that will go into Jessica and her team, and they'll get back to you on an ASAP. If you're watching this on demand, you can still ask questions. So the question box is still live. Any questions that you ask will be routed through to Jessica, and any responses that Jessica gives will come straight back to you in your registered email address when you registered for this webinar, or if you're watching it on demand, whatever you use to register will be the address that we send the answers to. So please do take our short survey. It will be on your screen. If not already, it will be there now. You can click the little pink icon that's in the array bar at the bottom and just do it now. It will take you no more than 30 seconds. And if you really enjoyed it today, please do use the share this webinar option. Just pop it around a few of your colleagues, because we really appreciate getting the message out about how great Zeptometrics is and how we can really help you. So that's really good. So again, thank you so much, Jessica, for your time today. It's been really good having you on the Untitled series. There will be more webinars. There's another one on December 9th this year, and we've got a whole calendar planned out for next year as well. So hopefully we look forward to seeing you again with us. And thank you so much for your attendance today. And don't forget, the on-demand link will be with you in about an hour's time. So please look out for that. Thank you so much, Jessica.
Thank you.
And thank you everyone here for watching. Take care. Be safe.